Okay, spermatogenesis. So we talked about the anatomy, and we talked about the fluids, and we talked about why that's important. Now let's talk about those gametes and the specific names of those gametes as they go through meiosis. So that's spermatogenesis, or the formation of sperm. And what I would recommend you do is pause the video and recreate this picture if you can, or if you want to add to this picture, but we're going to add quite a bit to this picture. So duplicate, I'm going to move it down here. And I'm going to go ahead and erase this stuff over here so that we can remember that we are going from spermatogonia into the luminous spermatids. Okay, spermatogenesis begins at puberty and will continue throughout the lifetime. We're making sperm. This takes about 64 days. We go through a couple rounds of mitosis to make enough spermatogonia. Then we go through meiosis to make spermatids. And then spermatids need to mature, so they go through spermiogenesis. Let's go ahead and draw this out on the right. I'm going to move this over to the left so we have more room. And let's go ahead and begin with, I'm actually going to move this a little bit more to the left. Yep. Okay, let's begin with spermatogonia. Okay, so spermatogonia, they will go through, and I will leave some space in between each of these terms while I add more to this picture. Spermatogonia, then once they go through their rounds of mitosis, then they become primary spermatocytes. Primary spermatocytes, as you can imagine, then become secondary spermatocytes. And secondary spermatocytes then become spermatids. And spermatids become sperm, which are actually the fully matured, or the almost fully matured version. If the sperm travel through the female reproductive tract, then they become even more fully matured. So by matured, we mean they're able to fertilize an egg. Okay, so let's go ahead and add some processes that happen during this travel. So spermatogonia to primary spermatocytes, we went through a bunch of rounds of mitosis. And mitosis means that we had a 2N cell, a mother cell, and we made a bunch more 2N cells. And then those went and continued on so that we could make a bunch, right? Then the primary spermatocytes begin meiosis 1. So they will go through meiosis 1. Meiosis 1, if you remember, we start with 2N. 2N makes, and then it cuts itself in half, but we still have, again, 2N. Oop. Then we go through meiosis 2. And meiosis 2, we took those 2N cells and we divided those in half into those haploid cells. Okay, so now we have 4 spermatid. So we have 4 of them. Those are our haploid cells. That's what we're going to call those. And then those haploid cells, which let me draw them like this. So N, 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 N. Those spermatids then go through spermiogenesis and spermiation to get tails. So, well, actually, hold on. So first, they will, let me draw this back in blue. So this will make more sense whenever we talk about what's on the next couple slides. But spermatids like sticking together. We'll see an equivalent scenario with oogenesis. But they typically will stick together until this spermiation step, where during spermatogenesis, we will actually break those connections between the 4 so that they can obviously separate. The advantage of keeping them connected throughout this entire time is to help maintain the blood-testes barrier, which we'll talk about. It's also been shown to have an interesting effect for the epididymis picking the right spermatids to go, or the right spermatids to continue on through their journey, because by having this connection between each of the cells, allows them to fine tune what's going on in each of them. So like, let's say the first one has the best environment for it. The other three might give up their journey and help contribute more to that end because they know they might not make it through the epididymis anyway. So this bond needs to eventually be broken, and that's where we're doing it, is in spermatogenesis and the rete testes. So then we also add on tails. So that's spermiation. So once we break those bonds, then we need to add on some tails so that they're mobile. Okay, so now we have sperm. Sperm are traveling through the ductus deferens. And if they exit through an ejaculation and they make it through the female reproductive tract, they may go through a process called capacitation. And capacitation is essentially where we make these sperm really ready to tackle that female reproductive tract. And so I'm going to draw a terrible picture again of what we just drew here. And I don't know how else to represent this. So I just thought like making them have like superhero capes maybe, but I don't know how to draw like a cape without making it look weird. So they just have like superhero capes. They're like, wow, look at me. I can go and do things now. Yay. So that's capacitation. In reality, what this means is they just get added enzymes and sugars so that they can make it through the female reproductive tract. Just worry about knowing capacitation. Okay, so let's go ahead and draw this. And why did we draw it next to the seminiferous tubule over here? Well, because I want us to pay attention to what's happening at what stage. So mitosis is going to, what color did I do that in? Okay. Mitosis is going to happen in the edges of the seminiferous tubule all the time. Then we have meiosis 1 and 2. So let me go with blue. Meiosis 1. So as they go down into the, I shouldn't say down, but go towards the lumen, they go through meiosis 1. Then at some point, then they go through meiosis 2. And then they become spermatotids. So then those spermatotids actually enter into the lumen. Okay, so then we can break this off. And then once they leave there, then they go through the epididymis, and that's where this green is happening. So this is happening in epididymis. And then capacitation would happen in the female reproductive tract. Okay. All right, so this will be helpful because now we're going to talk about what that looks like under the microscope in images. So here on the left is the same picture we just drew, except in a cartoonistic depiction, and showing you where those dividing cells would be. Then if we zoomed in, it actually tells you the names of those dividing cells. So you can see primary and secondary spermatocytes moving towards the lumen. And then they change names eventually. You can also see that the spermatids on the right side are all clustered together, or they're all next to each other. Like I said, they need to break off that connection during spermatogenesis. And then you can see at the top we have spermatids with tails. And this is a little bit of a cheating picture because it tells you that it's in the seminiferous tubule, but realistically this would probably be closer to the straight tubules or even the rete testes, because you wouldn't see any of those tails of the sperm until you got to that point. So this is important because whenever you start looking at the slides for lab practical, you'll see like some of them look really similar to others, except now you see a bunch of tails in this one and you don't see a bunch of tails in that one. That's how you know the absolute difference between epididymis and seminiferous tubule. Okay, let's see next. Oh, so we already talked about this, but by adding a tail, they become motile, so they're able to move. This does not mean that they know in what direction to move. They're just able to move. And we'll talk about what their tails look like in a second. The process is capacitation. They become motile. They're getting some extra fluids, and they're gaining additional capabilities. So like I said, they basically get some enzymes and they get some extra sugars. All right, so back to this picture. And here, I stole a picture from the internet showcasing what the nurse cells and the Sertoli cells are doing. So the nurse cells, what the nurse cells and the interstitial cells are doing, excuse me. So the nurse cells are going to help with spermiogenesis, and they are promoting spermiogenesis. So they are taking whatever they are responding to the hypogonadal axis, and they are saying and they are saying, oh, okay, we need to make sperm. Cool, let's make some sperm. And I like this picture because it shows you the separation of where those primary and secondary and spermatids are versus where the two compartments are. So the nurse cells are also responsible for maintaining the blood-testis barrier. And this is similar to the blood-brain barrier in that we just want to separate out what is touching blood and what is not touching blood. But we still need oxygen to go through all of these rounds. We still need energy to do this. So we need blood nearby, but we don't want blood touching. So the nurse cells are going to help create an actual physical barrier between those two. And we also have two compartments in order to help make this barrier. We have a basal compartment, which is what's holding the vasculature. And then we have a luminal compartment, which is closer to the lumen, and that is holding on to the developing cells. So the nurse cells are going to help maintain this barrier. Additional features of the nurse cells. They support mitosis and meiosis, like I said. They are responsive to FSH and testosterone. They will add nutrients and chemicals into the seminal fluid to help continue spermatogenesis. They will eventually start secreting inhibin, and inhibin is what is telling the rest of the body, hey, we're done making cells, we have enough, let's go ahead and stop the process. So this is promoting negative feedback back to the hypothalamus and the anterior pituitary. Nurse cells are also going to secrete androgen binding protein. Androgens, so androgens are typically testosterone, that's the main androgen that we talk about. ABP for short. Androgen binding protein is important for testosterone to actually get into the seminal or into the luminal compartment to do what it needs to do. Testosterone can't enter this environment without androgen binding protein, so it makes sense that the nurse cells are going to provide that for us. And of course, FSH, again, is helping do this as well. So nurse cells is super important. We're producing a lot of things that are really important to help promote the production of these cells and the maturation of these cells. And we're also helping separate these cells from the blood barrier. Okay, sperm anatomy. Once we're finally done with it all, this picture I made from the app Complete Anatomy. We have a head, a neck, and a tail. And the acrosome is surrounding the head. And the acrosome contains a bunch of enzymes, which your textbook and most textbooks say is essential to fertilization. This is actually a lie. There have been some studies that have shown that sperm without an acrosome are still able to fertilize eggs. There's something called an acrosome reaction, which you might have learned in high school. And this is where like the enzymes get released and then they dash their way through the layers of the oocyte, and then they finally are able to get to the membrane and then put in their genetic material and woohoo. But it actually turns out that if we removed all of this, the sperm cells would still be able to do that. They don't necessarily need that reaction to happen in order for fertilization to occur. So I think there's more to be studied here. We just don't know enough. But the acrosome is where those enzymes are. There might be other functions of it that we just don't know. Then we have the neck in the middle piece, which here is in like a teal color. And that is contains a bunch of microtubules and mitochondria. So mitochondria are going to be deep. They're going to be deep into the structure. They're going to provide a bunch of ATP. And ATP is going to help move that flagella, or flagellum. Sorry, flagella would be plural. And this is super important because this is how the sperm are going to help move through the relatively mucousy environment of the female reproductive tract. The way the flagella turns is in a whip-like corkscrew motion. So you can imagine what that would be like. And the mitochondria are going to help propel this motion. Why do we have microtubules? So we have microtubules at the end of the mitochondria towards the tail. And these are stacked in a way similar to like centrioles that you learned during mitosis. Not quite in the same formation, but relatively similar. And there's all of these specialized bonds in between them. Actually, let me kind of draw this out. So if this is like a cross section of the midpiece, and we were to look at the orientation of the microtubules, they would kind of be like this in a circle. Oh gosh, kind of like an aperture would be. And now I'm doing a terrible job of this. And they're in pairs. So, okay. And then they have these bonds between them. And depending on the orientation of those bonds will allow this whole thing to either rotate will allow them to rotate in one direction. And in order for them to rotate in one direction and change those bonds, you have to have ATP. So that's why the mitochondria are right there. ATP is going to help change the orientation of these bonds, and then that makes the microtubules move, which makes the tail move in that corkscrew-like fashion. Woohoo! Sperm.